Bismillahirrahmanirrahim. I'm Rafit Mustafa. Today I'll discuss type 2 hypersensitivity. Previously have uploaded a video on type 1 hypersensitivity. Now today we'll discuss about type 2 hypersensitivity. First, there is a synonym of type 2 hypersensitivity. This is called cytotoxic hypersensitivity. In type 1 hypersensitivity, we have seen that the antibody is fixed and antigen is free. But in type 2, it is totally different. Here, antigen is fixed and antibody gets free. So in type 1, there was antibody fixed, but here antibody will be free and here antigen will be fixed. Now, what are substances required to occur the type 2 hypersensitivity? First one, the antigen. Second was the antibody. And third one is the complement. In type 1, we have seen the antibody involved is only the IgE, but in type 2, the IgG and IgM will be involved, because we know that to activate the complement, only the IgG and IgM can only activate the complement. Not any other class can activate the complement, only the IgG and IgM. So here the antibody involved will be IgG and IgM. Now, if you see the mechanism, here we say there is a RBC, suppose it's a cell, and on the RBC there are two antigens. The complement, we have told that the antigens are fixed. Then a free antibody will come and attach with the antigen, and then an complement will come and causes a lysis of the cell. If we draw a picture for more clear understanding, suppose this is a red blood cell. It has two antigen on its surface. We know about antibody that antibody is a Y-shaped substance. Its head end is called FAB region and the tail end is called FC region. FC. Now the antibody will come and attach to the antigen. The FAB region will attach to the antigen. When the FAB region will attach to the antigen, the FC region will get activated. And when the FC region will get activated, complement protein will come. We know that complement are proteins that they take part in immune system by various mechanisms. When I will discuss about complement, I'll give a broad lecture on it. But for now, better understanding, we know there are 20 complement proteins, but only 9 proteins take part in the activity of immune system. First, first the C1 protein comes. C1 protein comes and divided into two subtypes. One is the C1A. Next, another one is C1B. The C1B take parts in the complement pathway, not the C1A. They get detached. Then comes the C4. C4 again, is like the previous C1, they get divided into C4A and C4B. C4B will remain. Similarly, C4, then C2, C2B, then C3B, come, then C5, it will divide into C5A and C5B. The next thing it will occur that C5B will activate C6. C6 will not be divided into C6A and C6B. It will remain solely as C6. Then C7, C8, C9. The C5B, C6, C8, C9. This togetherly is known as membrane attack complex, shortly told as MAC. So if we erase the picture and again draw it, what will happen here? First here I have told that C1 will come, it will remain as C1B, then it will remain come C4B, C2B, C3B, then C5B. It is C5B. Then it will cause it will attract C6, C6, C7, C8, and C9. This C5B to 6, this C5B to C9 is called membrane attack complex. Now the membrane attack complex will attack the red blood cell, and it will cause lysis of the red blood cell. So mean RBC death. This is the mechanism of type 2 hypersensitivity. How type 2 hypersensitivity occurs. Now, if we go some future forward, that what are the examples of type 2 hypersensitivity? First one, hemolytic anemia. I have told how hemolytic anemia can develop. Now, ABO transmission reaction the same way. RH come in hemolytic disease. Now, drugs like penicillin, quinidine, and hydralazine. Remember how penicillin can cause type 2 hypersensitivity. The metabolites of penicillin can attach to the red blood cell membrane and it will cause lysis of the RBC. How quinidine works? Similarly, quinidine metabolites attached on the platelet and it will cause lysis of platelet and it will develop thrombocytopenia. Next, the hydralazine. Hydralazine, its metabolite attached on the surface of cell and it will cause lysis of the cell and it will cause features like SLE. And then certain infection, mycoplasma pneumoniae. Mycoplasma pneumoniae antigen will attach on the surface of red blood cell and it will cause lysis of RBC. Now the rheumatic fever. It's very much important for the exam and also for practical life, how rheumatic fever causes type 2 hypersensitivity reaction. In rheumatic fever, when it is caused by group B streptococcus, mainly the streptococcus pyogenes. When the streptococcus pyogen enter our body, it has a protein called M protein. It's a protein called it has a protein called M protein. Against the M protein, the antigen is against the M protein is the antigen, and antibodies formed. M protein antibodies formed. But the problem is that the structure of M protein is similar to the cardiac myosin protein. Cardiac myosin. Cardiac myosin protein. It is similar structure. So the antibody produced against that M protein, and the antibody will attack the heart muscle, the cardiac myosin protein, and it will cause damage of heart valves, and it can cause pancreatitis. That means it cause endocarditis, myocarditis, and pericarditis, and cause damage of the heart. And next one is a good posture syndrome. In good posture syndrome, we know there are typical features. There are features of alveolar hemorrhage, and it is alveolar hemorrhage. It causes that it attacks the collagen membrane of lung, and it also causes the attacks the basement membrane of kidney. So there will be hematuria and hemorrhage. Thank you. If you have learned something, please like the video and subscribe the channel. Thank you.